I'm going to move on to introduce our next speaker, who is Matthijs Welkers, who will be speaking about monkeypox virus whole genome sequencing using NextGenPCR and Oxford Nanopore.
I hope everybody can hear me. Let's see where I can start. PowerPoint. Okay, good afternoon or morning to everyone. My name is Matthijs Welkers. I'm a medical microbiologist from Amsterdam, and I work at 2 locations. My day job is basically a clinical virologist at the academic medical center in academic virology for admitted patients. And then the other half of my time I spend at the public health laboratory, which is part of the public health service in Amsterdam, doing basically genome sequencing and genomic surveillance within our municipality. For those of you who don't know where Amsterdam is or what we sort of encompass within our municipality, this is viewed here. So we highlight it's quite a big area. It's 200 square kilometers, and we have about 890,000 inhabitants. And what the public health service does is doing currently all of the SARS-CoV-2 diagnostics that's free of charge for those with people with complaints. And the public health service also has a big sexual health center where anybody with complaints of STDs can be freely tested. And that we also have very good connections with, of course, the gay community then within our city. We're also a very international city with an airport close by. And as sort of a response to the previous speaker, we also did wastewater monitoring and found our first wastewater monitoring station that was positive was connected to Schiphol Airport. So that was one of our first things that we did. Now, how we do genomic surveillance within the region is via the Argos Consortium, which is a collaboration between our public health service, specifically public health service, the laboratory that does the diagnostics, and the Department of Infectious Disease, who does all of the source and contact tracing and goes to people, companies where there are outbreaks of notifiable infectious diseases. And from the academic medical center, we provide basically the academic part of it, so doing the genome analysis, and we also teach basically our source and contact tracers about how to do proper sampling, how many individuals do you need to sample from an outbreak, etc., to get as informative as possible genome information, and also give us the tools to do the proper analysis to provide for an answer for them to actually have meaningful public health actions and not sequencing as part of sort of a stamp collection. What we do currently for SARS-CoV-2 is also report on Twitter most of our variant analysis. So whenever we have some interesting stuff, we post it on Twitter as well as part of open data sharing as well. At the same time, we also identified monkeypox in Amsterdam. So when the first reports from England came, we were starting to get aware and immediately went to the sexual health clinics to ask if they also identified patients with symptoms that they could not really explain as part of gonorrhea or chlamydia infections or syphilis. And immediately they identified a few patients that they said they did not appear the same as what they have known. And we retrospectively tested those patients immediately for monkeypox, and already immediately sort of identified one from a visit from the 10th of May. So we reported that on the 20th, and that was the first reported case immediately for the Netherlands. And here you see sort of the outbreak part on the number of cases that we've had in the Netherlands reported to the first day of illness. So that's why it's previous to 10th of May in this slide. And within the Netherlands, we are sort of the main supplier of our monkeypox cases. We have, to this point, over 50% is all basically Amsterdam oriented or have been diagnosed within Amsterdam. So we are sort of the main center for monkeypox. Now, at the same time that this was occurring, we were trying to optimize our SARS-CoV-2 sequencing protocol. So we normally use Nikki Fried's midnight protocol, which takes about 3.5 hours. And we had a company that had this PCR machine called the NextGenPCR, which was basically a really fast way to amplify our genetic material, which took it down from 3.5 hours to 25 minutes. And since monkeypox was such a big genome, we really basically said, well, if it works for SARS-CoV-2, why wouldn't it work for monkeypox? So let's just, make an amplicon scheme and try it out, see what it does. And that's basically how it got started. And we designed together with other clinicians from Denmark, Martin Schau Petersen, who already had a sequence from the first monkeypox case using metagenomics, designed sort of the amplicon based, and the average size of the amplicons we chose to be 2.5 kb, just to get an average number of doable amplicons, else you'd have an enormous amount of primers in two reactions, and chose basically 2.5 size amplicon with the 200 base pair overlap. And as soon as we had our first successful amplification, we shared the protocol of it via protocols.io with a link here that we can share. Immediately, we also figured, well, it's nice that it goes on a fancy machine, but I think most of us will want to know whether it also works on the standard PCR that everybody uses. So we tried to validate it on the NextGen as well to have a really fast response, and also that if indeed this outbreak would become much bigger, we'd also have a way to quickly amplify and sequence a lot of samples. So we went about it in two ways. So one of the NextGen protocol, which ended up to be then an amplification of 42 minutes, and using a standard SARS-CoV-2 PCR protocol using the Q5 polymerase. And the amplification scheme is identical to that used for the midnight primer protocol using Q5, which is 3.5 hours. And after that, we sequenced it using the rapid barcoding kit on a nanopore. And we started with about 15 samples per flow cell, but I'll show you that you can even probably increase the number of samples per flow cell. The bioinformatic analysis that we did was quite standard. So the Minnow software used high accuracy live base calling. We removed the aberrant reads that were probably combined reads. Looked at the FOSS-QC at the raw data to see if it's okay, mapped it using Minimap, removed the human reads from the sample, and created coverage and consensus sequences. And at the same time, Cornelius already put from NextStrain a monkeypox build online that we used for the phylogenetic analysis as well, which is a very, very useful tool, and also NextClade. So we use that daily. And immediately within the amplification, this was our first attempt. And we immediately saw that we could rapidly amplify the entire genome quite well. There was only two amplicons which showed a relatively low coverage, amplicon 22 and 50, which appeared maybe that the forward primer of the amplicon 22 and 50 formed sort of a heterodimer. So the way what we did was just doubled up on the primer concentration in both amplicons, and that sorted out the problem. Combined with that, you need a relatively high input load for the PCRs. We next did sort of a dilution experiment to see what the sensitivity was, and to then see what kind of clinical samples do we need to select for successful amplification and sequencing. And what we did was taken a blister fluid sample with a CT value of 16 in our diagnostic PCR, made a dilution series to see what dropouts we would have with the amplification protocol, and then compared the standard Q5 SARS-CoV-2 midnight PCR protocol to NextGen. And you see that even in big dilutions up to CT28, we have, if your cutoff is about 30 or 35, we have a full genome coverage in one go using the Q5 one. So, and with NextGen, you see that within, if you have high loads, so average CT values of 1920, you immediately also get a full genome coverage. But there are, when the load starts to decrease, specific amplicons start to drop out, most likely because it's such a fast PCR protocol, and it's very finicky when you have such a big multiplex PCR that certain amplicons will most likely start to drop out. For the usability of the amplification, we noticed this is a combination of all of our diagnostic PCRs that we had. So these are from patients' combined samples. We had blister fluid, anus sample that we've taken, and throat samples. Throat samples have, in general, a very low viral load, which you cannot really properly sequence a full genome out of it. Certain ones are relatively high loads, and those you can sequence. But in general, most blister fluids have a very high viral load that will amplify without a scratch, basically, and get a full genome out of it. So that makes it quite available. And how we did it or used it was we had a few cases within the Netherlands. One of them was a child, about 9 years of age.
Back to back kind of day it's gonna be.
Somebody on the background. So we had a child with an unknown source of infection right early on during the outbreak, and we were quite concerned with the stigma that would come over this child or the father, that it was sexual abuse that might have affected the child. And the parents also really wanted to help and say, well, you know, this is not the case, and can you show or highlight that there's an opportunity to show that there's other ways of getting infected with this virus, and can you identify whether or not this virus connects or is located within a cluster of MSM individuals or not? And it did not cluster, so it was completely separate within the phylogenetic tree. Also, the epidemiological studies that we did it quite extensively. Went to his schools, went to his sporting clubs, and all of his best friends were tested, all negative. We also followed up on serology from family members, which ended up being negative. So we could not find a good reason how he got infected. And that, at least our paper gained a lot of attention and hopefully reduced the stigma on that not every infection with monkeypox is related to MSM transmission at the moment. Similarly, at the moment, we have about 66 samples that we've sequenced from the outbreak from early on. And one of the interesting clinical things, of course, that we wanted to know is, is there enough phylogenetic information within the sequences to see whether or not you can find transmission pairs or perform an outbreak analysis on these sequences, or is it just random mutations that are incorporated, or do we see a lot of overlap on non-epidemiologically related individuals having the same sequence, because that makes sort of sequencing not so much interesting. And we found 5 very clear epidemiologically linked. So these were individuals who know that they had it from their partners. And you see all 5 cluster either by using finding identical sequences, or just very minimal one SNP away from each other. So it gives a lot of confidence that at least the sequencing is accurate. And we've also did several experiments to resequence the same sample. And from those experiments, we also either identified exactly the same sequence or found one SNP with it, most of the times up or near homopolymer regions, which are using the nanopore technology that we use, are always a bit tricky within the region. So within the phylogenetic builds that we do, we sort of exclude the positions which we know are sort of the extended part of the homopolymer regions. We also immediately performed sort of retrospective testing on our samples. So we identified, of course, the first one on the 20th of May, and then we looked back into visitors to our sexual health clinics, those who had complaints or had positive PCRs for other STDs, because those are the ones that we save at the time. And we found several new cases which were actually predated the one that we identified. And the earliest that we have now identified is the 6th of May. And we tested back even other positive samples as well, back to March of this year. So this was clearly the first case that we identified within Amsterdam. So we don't believe that there was extensive circulation within our MSM population prior to the beginning of May.
Just to let you know that we are at time,
Yep.
so if you can wrap up.
Yeah, because there's a break coming, so I won't keep everyone. So these are also our conclusions. So monkeypox virus can be sequenced very straightforward using this amplicon-based approach. You can use any lab that does now use the Q5 polymerase with midnight primers can quite easily incorporate monkeypox sequencing within the protocols. You can run it on the same plate, and it's easy. Anybody can do this. Two amplicons appear to generate heterodimers, so with those, the problem can be solved by adding additional primers, and increasing replicant or amplicon dropouts occur when you increase your CT values or low viral loads. That happened more in the NextGenPCR and much less in the Q5. So a lot of people we thank that work together with this from Amsterdam and municipal health services and companies. And if anyone wants to get in touch, these are the main people who do the sequencing, myself, Marcel Jonges, and Anton van der Oude, who works with NextGen. So happy to take any questions.
Thank you so much, Matthijs. I'm going to set people free. We have a break, but also you can feel free to stay and ask your questions. We'll resume in about 10 minutes or a little bit less than that now. And we will be back with some more speakers soon. Thank you all for attending.